Hello everyone. I hope you have gone through my previous video in which we were discussing how this structure is formed, right? So I would like to add one point to this structure that we discussed the formation, how the extraembryonic mesoderm is formed, how the coelom is formed, but we missed one point. That is really important, that extraembryonic coelom, this cavity which is forming, as you can see very clearly in this diagram, this does not extend into this part of the this part. So this part forms a connecting stalk where the extraembryonic coelom has not extended. This forms a connecting stalk. This will form the future umbilical cord. Now, right? Now, proceeding further, now we'll take into consideration this structure, that is the bilaminar embryonic disc. We call it as bilaminar because it has only two germ layers till now, the ectoderm and the endoderm. So epiblast and the hypoblast, right? Above it, we all know there is an amniotic cavity. Below it, there is a secondary yolk sac. Then, to understand the further changes, I need you to understand that it is an embryonic disc. So you can imagine anything that is round and flat, like a frisbee, anything. So what was available with me, I have bought this structure, right? So this is an embryonic disc. You can imagine it is well circular in shape. So the above layer, we can consider it as the ectoderm. The lower layer, this one is the endoderm, so ectoderm and endoderm, and still we do not have any mesoderm in between, so we call it as a bilaminar, right? This embryonic disc, till now there is no demarcation of which is the right and the left axis, which is where is the ectoderm, where is the head end, where is the tail end of the embryo. So now what happens, there is the formation of a procaudal plate. How does that happen? Now this procaudal plate will be formed at the head end of the embryo, and that will determine which is the head end, which will be the tail end, what will be the right and the left axis, right? So what will happen, this is the endoderm. So suppose this is going to be the head end. Near the head end, not at the margin, near the head end, few cells of the endoderm will start becoming columnar, right? So if we just simply draw that, we can say that these cells of the endoderm, they will start becoming columnar. So they have become columnar. Now this structure forms the procaudal plate, and this is going to be the head end of the embryo, right? So in this bilaminar disc, this is the hypoblast. Few cells at the head end of the endoderm will start becoming columnar, right? Now, what will happen at the other end? So the other end will be the tail end. So what will happen at the other end? Few cells of the ectoderm or the epiblast now will rapidly multiply. And above there was the amniotic cavity. So when they will multiply, they will bulge into the amniotic cavity, right? So how will they do that? So this is the formation of procaudal plate. So this was the head end. Now at the opposite end or the tail end, few cells will multiply and form a heap, and there is an amniotic cavity above, so we say that they bulge into the amniotic cavity. So now this structure is called the cells. Now from the primitive streak, from this bulge, they will start going in between this epiblast and the hypoblast, right? So when they move in between them, they form the intraembryonic mesoderm. So we have the third germ layer, that is the intraembryonic mesoderm. This is intraembryonic mesoderm. So now this process is known as gastrulation. What is gastrulation? It is the formation of primitive streak, and from the primitive streak, formation of the third germ layer. Finally, the three definitive germ layers, that is ectoderm, mesoderm, and the endoderm. This is known as gastrulation. Now with the gastrulation, if we again take consideration of this disc. What did we study till now? In the endoderm, there is formation of the procaudal plate that determines the head end of the embryo. Then we have a bulge here, that is the primitive streak. On the opposite end, near the tail end. Now the cells will introduce themselves in between the ectoderm and the endoderm. What will happen? Now this disc is growing in size. This is a circular disc and gradually it will elongate to form a pear shape. Right? And as it elongates, what is the other change occurring? The primitive streak also elongates. Primitive streak also elongates. So what we can show is, like the earlier, the disc was a circular structure. Right? So we had a procaudal plate that determined the head end. So this is the procaudal plate and a primitive streak which was earlier a round bulge. This is the primitive streak, right? Now as the disc elongates, now it becomes pear shaped. And as it becomes pear-shaped, again, we have a procaudal plate. So this is the procaudal plate. And the streak now elongates, right? And from the streak, we studied that from the streak, primitive streak, the intraembryonic mesoderm develops. And that goes in all the directions in between the ectoderm and the endoderm. So this is the intraembryonic mesoderm, right? What else happens? This intraembryonic mesoderm doesn't go at 2 points. One is cranial, so we can say that this is cranial to the procaudal plate. This area is caudal to the primitive streak. So it won't go cranial to the procaudal plate and in an area caudal to the primitive streak. So at these two places, cranial to the procaudal plate, we will have an area where the ectoderm and the endoderm are in contact with each other, and this is known as this forms a membrane that is called the buccopharyngeal membrane. Similarly, caudal to the primitive streak, again there is an area where the ectoderm and the endoderm are close in contact with each other, because this mesoderm doesn't go in that area, and that is called the cloacal membrane. And because these membranes are so thin, they break down in future. The buccopharyngeal membrane breaks down to form the mouth or the oral cavity. The cloacal membrane will break down to form the openings of the anal canal and of the urogenital region. So this is how the embryonic disc appears, right? So in my next video, we will be discussing how the notochord is formed, right? Notochord, here I can just give an overview. What happens, there is a bulge that appears at the cranial end of the primitive streak. So this is the primitive streak at the cranial end of the primitive streak. We call it as primitive knot or the Hansen's node, and this is the Hansen's node which induces the formation of the notochord, right? So if you like my video, kindly press the like icon, subscribe my channel, and do view the next video for all the steps of notochord formation. Thank you.